Hello, everyone. Good morning or good afternoon. I'm Sabrina Buquois, Chief Marketing Officer at Inova Software. First of all, I would like to welcome you to our second therapeutic virtual partnering event on neuroscience, bringing together top neuroscience companies from across the globe for 4 days of online face-to-face meetings. This event is part 2 of the virtual partnering series Bio and Inova have decided to launch. We believe that there is a great need for more high quality and targeted partnering events and a need to unite specialized business development and scientific communities within specific therapeutic areas to bring the best science forward. Before we get started, just a few administrative logistics. Everyone's on mute to minimize background noise during the webinar. Please type your questions in the questions panel on GoToWebinar. And lastly, the webinar is recorded, so we'll send you a link to the recording. And it is now my honor to introduce you to Danielle Friend, who is Science Director, Science and Regulatory Affairs at the Biotechnology Innovation Organization. Danielle Friend serves as the Senior Director of Science and Regulatory Affairs at Bio, where she develops and advocates for policies that support the development of innovative therapies. Her portfolio includes work on issues pertaining to rare diseases and orphan drugs, pediatric drug development, and PDUFA and 21st Century Cures Act implementation, including patient-focused drug development, and she leads Bio's work related to the current opioid crisis. Prior to joining Bio, Dr. Friend was selected as a Science and Technology Policy Fellow with the American Association for the Advancement of Science. During her fellowship, Danielle developed policies related to genomic and scientific data sharing within the Office of the Director in the Office of Science Policy at the National Institutes of Health. Danielle received her PhD in neuroscience from the University of Utah. After completing her graduate work, Danielle conducted postdoctoral research at the NEH. Please join me in welcoming our esteemed moderator, Danielle Friend.
Great, thank you so much, Sabrina. Good afternoon to everyone, good morning, depending upon where you are. For those of you who are less familiar, Bio is the world's largest trade association representing biotechnology companies, academic institutions, state biotechnology centers, and related organizations across the United States, but also across the globe. Today, we're here to discuss the next generation of advances in neuroscience research with a specific focus on neurodegenerative diseases. In practice, neurodegenerative diseases represent a large group of neurological disorders that have heterogeneous clinical and pathological expressions affecting specific subsets of neurons in specific functional networks. Some of the most common neurodegenerative disorders include Alzheimer's disease, Parkinson's disease, Huntington's disease, and ALS, but there are, of course, hundreds of other neurodegenerative diseases as well. Today, we'll discuss current challenges in neurodegenerative disease treatment and the anticipated next steps in global research. Just before I introduce our esteemed panel, I do want to remind everyone that if you have questions throughout the panel, please send your questions through the chat function in the platform. That way we can get to your questions throughout the discussion. So with that, I'd like to introduce our 3 panelists today. First, we have Tor Christian Jensen, who is the head of research and development at PubGene. Tor Christian holds a master's and PhD degrees in computer science. He lectured in informatics at the Norwegian Institute of Technology in 1998 through 1999, and was a researcher in the Norwegian Cancer Hospital in 2001 to 2002. And a board member of 2 strategic programs at the Norwegian Institute of Technology. Next, we have Peter Levens, who's the Chief Commercial Officer at Alzheimer Inc. Peter joins us with his unparalleled depth of expertise in shaping Alzheimer's commercial landscape over 20+ year career in large pharma, but also rising biotech. At Pfizer, they partnered with Eisai and he launched Aricept, the first meaningful treatment for Alzheimer's disease. At Alien, he was the Vice President for Strategic Brand Management, and he led commercial development at of the first anti-amyloid antibody. As a group leader of Pfizer's US oncology marketing, Peter oversaw the launch of Suint, a breakthrough drug for renal cell carcinoma, He was also a senior vice president, head of late stage development. He led phase 3 and FDA approval of Onivyde, the first new therapy for metastatic pancreatic cancer in 20 years. Prior to Alzheimer, Peter served as senior vice president, strategic development, program management, and portfolio assessment at Tesaro, as well as managing R&D leadership, governance, and decision processes. He's a graduate of McGill University in Montreal with a BS in microbiology and immunology, and has a master's in business administration and marketing and corporate policy. And then last but not least, we have Harris Tzoulis, the vice director of Neurosystems Center for Excellence in Brain Disorders. Harris is a professor of neurology and neurogenetics. He's the director of research group on neuromics.org at the University of Bergen and vice director of the Neurosystem Med Center of Excellence for Brain Disorders at Auckland University Hospital. His research focuses on Parkinson's disease, neurodegeneration, and aging. With that, I am going to ask each of our panelists just to provide a brief overview of their organization and speak to the background and work of the work that they're doing in the neurodegenerative disease space. So with that, Tor, Christian, do you mind kicking us off for us?
Thank you, Danielle. It's very exciting to be here, and thank you for inviting me. As you said, I'm a co-founder and CTO, or head of research and development in Pubgen AS. We're a small bioinformatics company located in Oslo, Norway. As you said, I have a PhD in bioinformatics, and I've been working a lot with microarray and gene expression data analysis, and mostly applied to oncology and cancer research. And the key theme in my work, both academically and also with the company, is to combine information from text mining with regular or standard data analytics. In Pubgen, we have two main products. One is a biomedical search engine that is built on associations mined from biomedical text. It's called Corner Medical, and we also have a service for personalized cancer medicine. That is where we provide patients and doctors with up-to-date information about treatments and clinical trials. We base our analysis on biomarkers, clinical characteristics, and previous treatments, etc. And also, of course, a major component in that analysis is sequencing data. And our focus is to map possibly actionable mutations to targeted treatments. And we also have tools to analyze gene expression data, and also starting to work with proteomics, like mass spec data. In our technology, we have most of our tools are quite generic, and that gives an opportunity to move into the neurospace. And we started a collaboration with an Australian company called Genie US, and we're about to enter a collaboration where we're going to test our tools and methods to analyze some of their ALS data. So very excited to be here and looking forward to this neuro session.
Great, thanks so much, Tor, Christian. Yeah. And we'll certainly want to hear about some of those collaborations as we get a little further into the discussion. With that, Peter, do you mind giving us a brief intro of your organization and kind of what you're doing in the neurodegenerative space?
Happy to, Danielle, and thank you, Sabrina. Happy to be part of this panel. It's a really exciting time. You know, Alzheimer's is a very interesting and different company for many that we know. We are a clinical stage neuroscience company it was founded about 7 years ago by a gentleman named Martin Tolar. And really, the sole dedication of the company at this point is to develop the first effective disease-modifying treatment for Alzheimer's disease. That's really our goal. Martin assembled a team of individuals with great expertise, both in the pathophysiology of Alzheimer's disease, particularly understanding, deep understanding of amyloid protein misfolding and aggregation, and how that could be affected therapeutically. And also a group with very strong drug development expertise, having participated in major companies like Pfizer and others to bring important neuroscience drugs to market. So a real depth of science and late stage drug development experience. Our lead candidate is called ALZ-801. It's an oral small molecule designed specifically to inhibit formation of toxic soluble amyloid oligomers. And as we know, the science has emerged in Alzheimer's disease. They are really the key upstream drivers of Alzheimer's disease. We were very fortunate to recently have been awarded a $47 million grant from the National Institute of Aging to advance the program. And now our phase 3 trial in early Alzheimer patients is scheduled to start in early 2021. So a lot of momentum, a lot of excitement, and a lot of convergence of the science around the areas we've been focusing on. So excited to be here.
Great, thanks so much, Peter. And Harris, do you mind just giving us a brief overview of your organization and what you're doing in this space?
Harris, you're on mute. Would you mind? You'll have to repeat what you just said.
Thank you very much. Thank you very much for the invitation, and apologies for being late. I had some technical problems when I was trying to connect. So very good being here. My name is Haralambos. Harris is the short version, which many people who know me are very grateful for. It's a very complicated and long Greek name I was privileged with. I am a neurologist. I'm a professor of neurology and neurodegeneration. I work in Bergen at Haukeland University Hospital, and my main activity these days is, in the last few years, is research. I lead a research group dedicated to the study of Parkinson's disease mostly, and also I'm the co-director of a National Center of Excellence for clinical research in neurological diseases, with a main focus on Parkinson's disease, dementia, ALS, and multiple sclerosis. So our center, and I think I will speak mostly about our center because this is probably most relevant to this discussion and this forum. Our center is called NeuroSysMed. This is short for Systems Medicine in Neurology. And essentially what we do is we do translational research to identify targets and take those further into therapies and drugs. And then we do clinical trials to test those drugs on patient populations. At the moment, we have 2 big trials, academic initiated, so investigator initiated trials on Parkinson's disease, in addition to several industry initiated trials. We have 1 trial on dementia with Lewy bodies. We are planning 1 trial for Alzheimer's disease, and we have 1 big trial on ALS, and at least 2, I'm sorry about being imprecise here, multiple sclerosis is the responsibility of my co-director, Kjell Morten Mir, but I think we have at least 3 trials on multiple sclerosis. And of course, our main goal, our main aim is to develop and test drugs that can delay the development of these diseases in particular. So I'm director for neurodegeneration. That's where my main focus is, and in particular for those diseases where there's very little we can offer patients these days. That's our main focus, I would say.
Excellent, thank you, and thank you all 3 of you for joining us today. Well, why don't we go ahead and kick off the discussion? Historically, the development of treatments for neurodegenerative disorders and certainly brain disorders in general has been particularly difficult. I'm wondering if each of you could comment on kind of what you have seen as the biggest barriers for advancement for the development of some of these innovative treatments for various neurodegenerative disorders. Peter, would you like to kick us off?
Sure. I think there's sort of two big themes, in particular in Alzheimer's disease drug development. I think it's really first is getting a deep understanding of the biology of Alzheimer's disease, amyloid biology, and understanding what truly is the target for a therapeutic intervention in this disease. It's long been known that amyloid was important, that we had the amyloid cascade hypothesis, and that was sort of borne out for a long period of time. But as the therapeutic probes test the different elements in the cascade, there's really been this growth and understanding that it's really about intervening with the most toxic species, which is soluble amyloid oligomers. And the other big learning is that these oligomers, it's not just part of interrupting a cascade. where the event may be the development of plaque, but it's really to understand that there's a direct toxicity that's being manifest by these species on the neurons in the synapse that needs to be addressed directly. So that's a big thing, knowing the right target, and I think we're there now. And the other thing is, we didn't really have, until this point, we did not have specific and sensitive biomarkers for the disease. We understood amyloid, and we're looking at various measures of amyloid, but we really didn't understand what is the key process, what are the key call them disease progression markers that we could be following. And it's exciting that in the last 9 months or so, there's been a huge growth in that area. And a real sort of a renaissance of plasma biomarkers of disease progression, and I think that's going to really change and open up the field tremendously.
Great, and Harris or Tor Christian?
Sure, I can. I don't know. Is this completely open, or are you giving the word?
Please, yeah. Discussion among all of us. Yep. Please go ahead.
No, so I agree with Peter. Of course, I'm going to be a little bit provocative, perhaps a little bit the devil's advocate, and say, I don't think there have been major advances. I don't think there have been big enough advances. Of course, I agree with Peter that understanding the amyloid biology, understanding the toxic species, and so on, and advancing the understanding of protein deposition also in Parkinson's disease is important. However, on the treatment side, things have been quite disappointing, and really the whole trip of, the whole journey of understanding neurodegeneration has been going on for more than 100 years, and yet we have very little to show for. I think things may be going in the right direction or not, I don't know, but we still lack the big breakthrough that other fields, such as autoimmunity, inflammation, or cancer have had. We still haven't had this breakthrough, this or these breakthroughs. Whether these breakthroughs are going to be in terms of the proteinopathies, in terms of things like lysosomal dysfunction or mitochondrial dysfunction or anything else, I don't yet know. But I'm waiting for this big breakthrough that will make the paradigm shift and the change. There is a point of discussion further down, but the roller coaster that the amyloid antibody has been is just an example of that. It's taking a disappointing turn again, and we really don't know where it's going. So, and on the other hand, there have been developments into stem cell biology. Some of this has shown potential, but again, we haven't seen good results. And again, I know there is a lot of excitement in Parkinson's disease in terms of stem cell therapy at the moment. A lot of my patients ask, but my main concern is that even if we make this work in a way that is much better than the first tries when they tried with the adrenal medulla cells, and I think we will, because these are iPS derived, even if we achieve this, what guarantee do we have? How do we know this is going to be any better than DBS, the deep brain stimulation? And again, we will not do anything about the continued neurodegeneration of Parkinson's disease. We will only fix for a period of time the motor issues. So yes, I'm sorry for the pessimistic tone. It's more yes, I want to see a breakthrough. I haven't seen it. I haven't felt it yet.
Yeah, and I think you're right. And this idea of a breakthrough is always tough, because it might just be that it's the first incremental gain, that enables more direction or more investment or movement towards a particular avenue that looks promising. I spent a lot of time in oncology, and most of the gains in oncology were started by an incremental win, a small gain that then brought attention to a focal target and then produced a much larger benefit through multiple therapies and combination therapies and other types of things. So sometimes the wins are just behind the corner. And the other thing I think that's really important to understand is that sometimes the failures give us really important learnings that we have to capitalize upon to get the success that you're looking for, to get that breakthrough. So I would let's look carefully at what the failures are and understand them really well, because that might be the path to that breakthrough you're talking about as well.
Absolutely. Yes, those failures, as you call, yes, scientifically speaking, they've given us a tremendous amount of knowledge, and we need to capitalize on that. Absolutely. It's also the other problem, right? We're in the field of cancer. We were trying and are trying to kill something. In this case, we're trying to keep something alive, and there is a very big difference between these two things.
Absolutely. Yeah. Oh yeah.
Well, I think we just kind of alluded to this a little bit in that late last week, there was an advisory committee meeting here in the United States for the Biogen therapy aimed at clearing beta amyloid from the brain for treating Alzheimer's disease. And, you know, I think that it seems the FDA has really tried to partner with Biogen in, you know, thinking about their clinical trials, but unfortunately the discussion from the advisory committee meeting wasn't, you know, super positive. Certainly, you know, FDA can still make a different decision as they go into the new year, but wondering if you all could comment on kind of what you see coming out of that advisory committee meeting. You know, how does it impact Alzheimer's disease development as we see it now? What lessons learned can we take from it?
Yeah, I mean, I can begin if that, I don't want to, you know, go over other people, but, you know, I think as we watched it, it was really a remarkable event, okay? It was the most unusual advisory committee we've ever seen. You know, there were sort of three big themes that were coming from the advisory committee. One was, you know, the panelists, the invited panelists were very frustrated and upset, almost more by the process and what they were being asked to do than by the actual inspection of the data that was being presented by the sponsor. So there was that immensely negative thing that came. And part of that was, so that was one theme. And then the other theme that was startling, I think, for many people, but actually is so important, is that the FDA was really trying to collaborate with the sponsor to bring a drug forward in a way that we hadn't seen in the neuro division before. So it was really, it was a bit surprising, but it actually was really important because it shows that the agency wants to partner with other, you know, not only the, for example, the NIA and others to really move a drug forward. And that's genuine, you know, we shouldn't doubt that in these political times. And then, you know, the next was that from the commentary by the individual members, the sort of open comments by open speakers. And there we saw this completely reinforced the need for why we have to work with urgency in Alzheimer's disease. The Alzheimer's Association stepped forward and said we have to have a first step. We have to have a first step, even if that first step is imperfect. And then there were, I think, at least 6 Alzheimer patients, many of them APOE4 carriers, who really spoke to about the value of the disease and the importance of the disease. So for me, that was like, okay, this re-energized me towards let's take what we learned here and apply it and come up with better therapeutics, and then not throw out the baby with the bathwater. Understand what the learnings are from that. And the learnings that we took were that, you know, that an anti-amyloid therapy can be effective if it, in the case of aducanumab, if it's dosed high enough and sufficiently long to target the key toxic species, which is the oligomers. And then we also saw from Biogen that they've now understood their drug much better, and they've now elucidated a new mechanism, which is precisely an anti-oligomer mechanism of action for their antibody, which is distinct from the other antibodies. So those are 2 powerful learnings that we got from that, and, you know, let's apply them to the future therapeutics.
I think another factor is, and I think Peter mentioned this earlier, the biomarkers, and to Christian also mentioned a little bit about this, and this is the ability to detect disease early, because of course removal of the toxic forms of beta amyloid will, or should at least, be much more effective if it's done early enough. We don't know, I don't have any scientific basis for saying this, but, you know, several in the field believe that there may be a point of no return in these diseases. I don't know, I haven't seen any, you know, conclusive data showing that it actually is like this in humans, but it might as well be. And this drags into the discussion all the things that we are learning right now, the exciting properties of these proteins, the prion-like proteins, and all these things. So it may be that we do have a point of no return, at least given today's attempts to treat, and you know, starting early is extremely important here. but we need biomarkers to do that. And in
Yeah.
Alzheimer's has been much better than Parkinson's disease. It's not there yet, but it's getting there. I mean, CSF markers and nonetheless PET-based markers for amyloid. And I think there are some promising results coming on PET-based tau detection. In Parkinson's disease, it's been much more disappointing. So for some reason, alpha-synuclein hasn't had the same hasn't generated the same breakthroughs as beta amyloid in Alzheimer's. And also alpha-synuclein correlates much worse with disease severity and with neuroanatomical distribution in Parkinson's than amyloid does in Alzheimer's. So I think we're a bit more uncertain in PD than in Alzheimer's disease about the role of our protein in this disease. It's always there. We know it's at the scene of the crime. We know it's caught kind of, red-handed, but, has it actually done the murder? We don't know that. It's just there. It's always there, but that doesn't necessarily have to mean that it's the culprit. So
exactly.
Yes, I'm a skeptic there. I don't know.
I think
there could be another culprit lurking in the shadows that we haven't seen yet. Yeah.
Exactly.
Oh, and sorry, Tor Christian, I think you had a comment there.
Yeah, sure. To me, I think one of the key things this meeting has taught us is the complexity of the disease, and that we still have a lot to know about how to do the correct stratification of performing the clinical trials in order to get, or hopefully get, those results that you want. So the data will not be that conflicting or uncertain. So I think that is probably one of the lessons from this case with the aducanumab approval discussion.
Absolutely. Well, we'll see kind of, what decisions FDA makes as we go into the new year, but certainly I think there are a lot of lessons learned from this particular case. Maybe the next question I'll first ask Tor Christian to comment on, and perhaps Peter and certainly Harris, feel free to speak up. But we're seeing more advances. Personalized medicine can really make great advances, especially in the context of oncology. That's where we've seen a lot of work really tackle some of these cancers that have really, been a problem for people. How can we think about precision medicine in the context of neurodegenerative diseases? What do we need to do in order to get to a more precision medicine approach for neurodegenerative diseases moving forward? Christian, Tor Christian, do you want to start for us?
Yeah, I think we still need to have more information and deeper knowledge about the pathogenesis of these diseases and exactly how they develop and which factors that can actually be actioned upon. I don't think we have enough knowledge to fully realize personalized medicine in this area yet. If you look at oncology, where you have a number of mutations with specific targeted treatments that can actually make a difference, we are not there yet with the neurodegenerative diseases, I think. And yeah, that's at least my view on it as of today.
Great. And Peter or Harris, any comments, thoughts on that?
Yeah, well, I think so. Obviously, the genetic contribution is one of the key determinants, and it's led to, particularly in key mutations that are sort of deterministic of the tumor or the cancer type. Have been very, very helpful in oncology. But I think we should still follow those same types of leads in Alzheimer's in particular. That's the area I can speak to. And we see this one major risk gene, which is ApoE4, which is really contributing to individuals getting diagnosis of the disease probably a dozen years before the non-ApoE4 patient. And so that really gives us a strong lead, but in and of itself, it's not enough. That's not precision medicine. It's precision medicine if you have a strong biologic rationale or biologic hypothesis as to why that subset of individuals might be predisposed, and you should target them for your intervention. And so, in our case, the big understanding that we have in this disease that helps us target is that APOE4 patients, because they have a diagnosis of Alzheimer's disease much earlier, it's very important. It actually proves that these individuals is actually almost a diagnostic for having amyloid Alzheimer's disease. So a patient who has APOE4 will likely 98% of the time and almost 100% will get the disease by age 82. But what's the biology underlying that? And we see that from authors such as Hashimoto and others that these individuals also have up to 3-fold higher levels of toxic amyloid oligomers. So here's this connection between the genetics, the biology, and then the target that you want to intervene on. And we've used this as a key determinant of our approach clinically to focus our phase 3 program on these APOE4 patients because of those two features: high diagnostic confidence that they do have amyloid version of Alzheimer's disease, and also the strong biology supporting why a drug such as ours would work there. So that's that precision medicine you can apply to Alzheimer's in our case.
Great, thanks, Peter. Harris, anything you'd like to add?
Yes, I actually believe that the heterogeneity of these disorders of these diseases is a main bottleneck that's limiting us from making breakthroughs. So we call Parkinson's disease by one name, Parkinson's disease, but this is a clinical definition, and it's based on what clinicians see, which doesn't have a very high resolution, to be honest. And we know, as Peter mentioned, we do know that there is genetic heterogeneity, but even beyond the genetics, because genetic contribution is small in Parkinson's disease. It's there, but it's small. Even GBA would not be sufficient. But yes, there are pockets of populations, PD population, with concrete genetic defects, such as the GBA defects or the LRRK2 or whatever other genes are contributing, but also in the so-called idiopathic PD and even idiopathic PD without GBA variation, I believe there are subgroups. So a lot of our non-clinical trial-oriented research at Neuroscience is actually aiming at identifying pockets, subgroups of patients within the big group we call idiopathic Parkinson's disease, and then define biological pathways. I'm not going to say mechanism, because we know it's hard to say anything about causality there, but at least identify biological pathways defining or associated with each subtype, so that we then can start moving towards personalized trials, because I think it's essential. I think it's a main reason why a lot of the trials haven't produced results. And honestly speaking, it's a little bit tragic too, because sometimes I'm wondering how many of the neuroprotective trials in PD or corresponding ALS or AD, for that matter, may actually have worked for a subgroup of the patients exposed. And because this is not a tumor disappearing, if it worked in 5 out of 200 people, you would not see that. This would drown in the statistics of a phase 2 trial, simply because you wouldn't notice a slower disease progression by, I don't know, 3 UPDRS points in 5 people. You would simply not notice that. If it had been a cancer trial and the tumor had shrunk significantly in those 5 people, you might, the radiologist might, oh wow, look at that, maybe you want to look at that. But in neurodegeneration, we don't see it because it's mostly negative effects we're measuring. So it is extremely important that we take this into consideration. So we have in, I mentioned the clinical trials before, we have a big cohort study called the STRAT-PARC and a corresponding one called the STRAT-COG, which aim to collect a large number of patients and stratify them according to underlying biology. And these are starting now. So STRAT-PARC just started, and STRAT-COG is starting in 2021. But we expect a very high recruitment rate based on our national reach in the country and our international collaborators. So there we want to identify, and we want to do this both, not the old way. We don't want to just check for, GBA variation, LRRK2 mutations. We want to collect as much data as possible, in a sense, digitalize as much of the patient as we can in terms of genomic, epigenomic, transcriptomic, proteomic markers, and then also do a hypothesis-free, data-driven analysis to identify clusters there. We have preliminary findings suggesting that there may be some subpopulation there in Parkinson's disease, which I'm not at liberty of discussing right now, but in terms of metabolism, for example, we do see some subpopulation, so that is interesting. So I think that is essential to identify those subgroups and then to make clinical biomarkers that can select those and that can monitor those under clinical trials, I'm sorry.
Right, absolutely. And I feel like we keep coming back to the conversation of the need for really strong biomarkers kind of across neurodegenerative disorders so that we can make sure we're identifying patients, diagnosing them correctly, being able to track their disease progression and certainly treat them as well.
Even just making the diagnosis, you know, I can mention a fun fact. We have one of the oldest Parkinson cohorts in Norway, which I'm involved in. It's called the Park West cohort, and it's 200 individuals with Parkinson's disease and their spouses as controls, followed up since 2004. Now these people, okay, 200 people with Parkinson's disease followed up since 2004 systematically. This is a research project, and followed up by the country's foremost experts, right, in movement disorders, PD experts even. And now, because it's since 2004, we're now accumulating a collection of brains because we have a brain bank, and we have about 50 brains collected at the moment, out of which 2 do not have Parkinson's disease. So, you know, even diagnosing them, I mean, 2 of them have no nigrostriatal degeneration and no alpha-synucleinopathy, and it's, you know, clearly these people had been followed up by PD experts systematically as part of a research project, and yet, so, oh, we need biomarkers.
Absolutely.
Yeah, and I want to reinforce that point that you made about having a multiplicity of biomarkers. You know, the proteomic information, the patient, even the patient history, even the patient trajectory, the you know, the genetics all together, because, you know, we're dealing with systems diseases. We're not dealing with singular, you know, a single cell, a single site. We're dealing with diseases of, you know, the microenvironment of the neuron, for example, in the brain. So we have to think about gathering all that data from all the sources we can, and using the big data principles that we have to understand the full systems, create a systems model or systems view of the disease we're targeting. I think that will really help, and that, you know, that's great. You know, the great advance we've had recently in Alzheimer's disease is that we now have a lot of confidence from the labs of people like Kai Bleno and others, and Eric Ryman of the Banner Institute, showing a path for, you know, a plasma biomarker, p-tau 181 or 217, you can take your pick, but that are really, really good. And they're really good. They're almost, you know, acting more as theragnostic biomarkers that we can then apply in these trials to monitor progress, to understand how the biology is changing, you know, subject to an intervention, which is so powerful that we didn't have before, you know. And so we have that. And then we also have, I think, a lot of progress, and people haven't quite gotten their arms around it, is the structural biomarkers, the imaging biomarkers that we have in the disease. So now we also see that if we study the structure like the hippocampus in Alzheimer patients, we can see real changes, and we can track those changes in those patients really well. And so now we can use the clinical endpoints, we can use the fluid biomarkers, the CSF biomarkers, and we can use the imaging information and use it in totality to really get a great picture of what's working and what's not. So I'm incredibly optimistic right now.
Great. One thing that I think Harris alluded to was the size of clinical trials that are often needed to be completed for some of these highly prevalent chronic neurodegenerative diseases such as Alzheimer's. Peter, maybe you could kick us off by answering this question, but you know, we've heard a lot about companies, small companies in particular, having a hard time, you know, with the cost associated with some of these larger clinical trials that need to be conducted. Can you comment on kind of how companies are addressing this barrier? Is there an opportunity for partnership there in order to make sure that, you know, some of these small, really innovative companies can be working in the highly prevalent chronic disease space?
Well, absolutely. There's always, you know, there should be collaboration. That there's been a lot of data sharing around placebo arms of trials and contributing that to the common good, which has been very useful. You know, there's the concepts of shared control arms in trials. I think one of the biggest contributions a small company can make is getting back to the biomarker and the precision medicine approaches to focus on the patients in which you predict a meaningful or a larger effect size. and allows you to run a study in a smaller population of patients that are homogeneous, and therefore you can have less of the sort of variability that you'll see or less noise in the signal, and that permits you to run a more effective, faster trial and smaller trial. And then there's the economies that are generated by having meaningful biomarkers, so that you can then empower the clinical output with biomarkers to correlate. So you have a sort of a translational and a clinical perspective on that. And that permits you to run a smaller trial more efficiently, more quickly, and that all has great benefits. And that would be a huge change from the previous time, where we felt we didn't really understand a disease enough, so we mounted a massive trial, 1,000 patient trials in Alzheimer's disease, followed people for an incredibly long period of time. Didn't see much signal because there was so much, even despite the size, there was so much noise in the system. So this is a new era. I think that small companies can be really good at understanding their biology, understanding their mechanism, and use that as their most powerful weapon for running at the right trial.
I agree with Peter completely on this, and I think small companies can also look into collaborations with academic organizations. There aren't many of these, so this is in a sense, there is a saying. I come from Greece, and I left when I was a teenager, but some things get imprinted because I grew up there as a kid. And there was a saying that you have to say something good about your own house, lest it falls on you and kills you. So So just to say something about NeuroSysmed, our center, we are very interested in this type of collaboration. So we are a nationally funded center of excellence for clinical research, and this is about 32 million euros from the state for a period of 8 years. And of course, in addition to this base funding, which we use this funding basically to build our backbones, our labs and our clinical trial coordinators and our nurses and all that. And in addition to this, of course, we're applying all the time for extra funding for the individual studies we're running. But I think centers like these also, as we are trying to do, can collaborate with the industry, including and perhaps especially with small companies and startups, because they don't have the bulk, they don't have the muscle bulk, they don't have the wealth. They're not like Biogen, like Novartis, and to common projects. And for us, we don't ask for equity in these companies or anything like that. We're an academic institution, but of course we want to promote science. And we have built a one-stop shop, as we call it, for clinical trials, meaning that when a company comes to us and wants to test something, we make it really easy and quick to get the approvals, to get the paperwork, to appoint the clinical trial coordinator, and so on. So I think industry and academia need to work together more, and that can lift and promote the startup companies.
Great, excellent. And I think you just alluded to, one of the collaborations we kind of wanted to talk about on the call today. We certainly have good representation from the three of you kind of across different sectors that can, certainly partner to help us better develop treatments for neurodegenerative disorders. I'm wondering, Harris, beyond what you just said, Tor, Christian, or Peter, if you have other thoughts on some of the innovative collaborations you've seen in the context of developing therapies for neurodegenerative disorders.
Well, go ahead. Sorry, go ahead.
Go ahead, Peter. It's fine.
Well, so obviously one of the biggest collaborators we have is the National Institute of Aging. They are great, they're passionate scientists. They have a real platform to support all the elements of the development of aging illnesses. And I would take a look. They have a whole series of grant mechanisms for small companies and small grants, early stage development, data as well. So that they're great collaborators. And then we've seen, among the Alzheimer's community, I mentioned the labs of Kai Bleno and others. They really, really they understand their part of the puzzle really well, and they're more than willing to work with anyone and everyone to help share that knowledge. So those are great collaborators that we've seen. I mentioned the Banner Institute, Eric Ryman, those type of people as well.
Great, and maybe Tor, Christian, I'll ask you kind of how do you see companies partnering kind of on the data side of things?
I think data sharing is very important in this area. I've seen like in the cancer community, there are huge international collaborations working together to share data, and I've seen a tremendous amount of publications by not the original authors, but providing interesting and good results on others' data, and I think that is an important way to go. But I think to pick up on what Peter just said, I think government-funded initiatives and agencies has to play an important role now in this area because of the protracted disease courses that are so common, and especially if you're looking for early detection biomarkers. It's really difficult for small companies to fund such long-term projects that is absolutely need in order to get the good or the relevant biomarkers for early detection.
Great, thanks so much. So we are coming towards the end of our time here. I'm not quite seeing questions from the audience yet, but maybe some questions will come in. I think, one of the last things I wanted to hear from you all, I wanted to end on kind of a positive note. Thinking about, what do we imagine seeing in the neurodegenerative space in the next 5 to 10 years? And how do we get there? I think we've raised some good points already today. Specifically biomarkers has come up a lot, and, really leveraging biomarkers as we go forward to help us develop some of those innovative treatments. But again, turn it to the panelists. Where do you see us in the next 5 to 10 years? Kind of optimistically, hopefully, in the space of developing treatments for neurodegenerative disorders, and how are we going to get there?
Well, I would like to see a patient with PD or Alzheimer's or any other neurodegenerative disease coming to the hospital, getting a diagnosis, and then getting a complex biomarker package, and then being told, you have a form of Parkinson's disease. It is form XY. And we know that when you have this form of Parkinson's disease, this is how it's expected to develop, and these are the biological mechanisms that are important for you. So currently, I'm not going to say we have therapies because 5 to 10 years is not enough perspective. Currently, we're running 1 or 2 trials aiming at exactly this area, and you can enroll if you want. That's what I would like to see. The patient coming in, being diagnosed, and then being stratified, and then being canalized to appropriate trials in each targeting each biological mechanism.
Great, and Peter or Christian?
Well, I think so. 5 years from now, right? So even I think that seems like a long time, but it's really not. And I think that there's some really exciting developments. I think first of all, just the development of understanding a patient's genetic and proteomic profile can really help. So I see an environment in 5 years where individuals who have family history of Alzheimer's disease or the genetic profile that predispose them to get profiling for the biomarkers that are indicative of elevated risk of disease, and those are available. You could go into a doctor in a few years, and maybe even less, and say, I need a plasma p-tau test to see if I'm at an elevated risk of progression to Alzheimer's disease while I'm asymptomatic and still working and still living independently. That's very possible. I also see the advent of wearables and big data application of information that we're using, computer use, and other subtle changes that could be tracked in individuals, and then driven through big data analytics to show that individuals are exhibiting signs that are so subtle that are indicative of potential future cognitive decline, and that could be another great enhancement that we could bring in those years, so that we could have really getting to the front edge, so that we're intervening in people that are healthy and independent and living well, and avoiding having to deal with the situation when they're in the middle of a florid disease and there's not much you can do. So I think it's just going to be it's going to be a real renaissance, more than a renaissance. It's a whole new era in 5 years.
Great, thanks, Peter. And Tor Christian?
I think generally what I'd like to see is that we get further on understanding the mechanisms of the disease development and be able to detect the cases earlier. And definitely, of course, we're not near a definite treatment for any of this, but I really like to see that we advanced far enough so that patients can have a realistic hope of a near-term treatment option.
Great, great, excellent. Well, we do have one question that came in from the audience, so I'll pose it to the group and see if there are any comments or reactions to it. But the individual said, I wonder whether the amyloid beta oligomers aren't the end of previous pathophysiological drivers, such as antioxidants, inflammation, and other dysregulations. I don't know if anyone has any comments or thoughts on that.
Well,
Yes.
our chief scientific officer would say that that they are the beginning. They're the upstream drivers, and the consequences of that were mentioned in that case are actually downstream of those. There's a whole multiplicity of changes that are unleashed by the accumulation of soluble oligomers, and but they are definitively in test situation after situation. We see that they predecess most of those other changes, and so therefore I would say it's probably the opposite, that the oligomers are upstream.
Okay, great, thanks, Peter. Well, I think those are the questions we had for today. I do want to thank you. We appreciate you joining us today, Tor Christian, Harris, and Peter. It was a great discussion. So thank you so much. And with that, Sabrina, do I turn it back to you, or are we all set?
I'll take the floor for for just a few minutes. So I wanted to thank you very much, Tor Christian, Peter, Harris, and Danielle, for this great session. Just a final word to let everyone know that there will be one last virtual partnering event before the end of the year, and it will be focusing on infectious diseases. And registration is now open at virtualpartnering.com. As we close this webinar, we would really appreciate your feedback. So if you could take 1 minute to answer the quick survey that will pop up on your screen, that would be fantastic. We would like to thank you all for attending today and wish you a great event with lots of new connections and collaborations in the neuroscience field. Stay safe and stay healthy. Thanks, and bye-bye.
Thanks, everybody. It was a pleasure.
Thank you.
Thank you, panelists.
Take care.
Thanks, Danielle. Thanks, Sabrina.
Cheers.
Bye.